Thank you.
Thank you very much, and thank you very much for the invitation to talk today. So I got the easy bit, to talk about new therapies and new directions. And what I'm going to do is I can't cover everything possible, but I'm going to mention some different classes of drugs and how they work. And then I'm going to take you in a different direction, looking at some of the new technologies, which may or may not eventually come to the MPN field. So the first drug class that I would like you to consider are the telomerase inhibitors. And these are drugs which act, we're going to see a lot in this, of the process of DNA making RNA, making proteins at the cellular level. And the telomerase is required for this process to allow the string of RNA to be made. And telostat is a telomerase inhibitor. It is an entirely new class of drugs that have never been used. So it's what's called a first-in-man drug, where it's brought in completely new mechanism of action. These drugs were in cell studies were shown to stop replication of cells, particularly cells called CFUMK, which are megakaryocyte precursors. In other words, the cells that make lots of platelets. And in cell line studies, you can see that these drugs inhibit the growth of CFUMK. So that suggests that these might be a very useful drug for treating of myeloproliferative disorders. So that's one drug that is currently in clinical trial in the EMBARK study. An entirely new type of drug, totally different from all the things that Professor Harrison was talking about. So telomerase inhibitors are one class of drug. This is another entirely new way of looking and thinking about how to treat MPNs. So the problem is the fibrosis and myelofibrosis. Is there anything we could do about this? Well, the pentraxins are drugs which look at the repair of the fibrotic tissue. And what they do is that these drugs in green latch onto the damaged tissue and then the cells and basically block the fibrocytes which make the fibrous tissue. So the idea would be that if you use these drugs, that you might get reduction in the bone marrow fibrosis and therefore would restore the normal hematopoiesis and improve the cytopenias. Obviously myelofibrosis we're talking about. So these drugs, again, a totally different type of drugs, are being used again in clinical trial. The PROMOTE study is ongoing. I don't have any answers to whether these actually work or not yet, because the trials are all actually testing them to see if they work. So that's two different types of drugs. Another different concept that is also coming into clinical trial are these drugs. These drugs all have great names. Luspatercept, and the other one, which is almost the same, is Sofaracept. And these drugs work in a different way again. What they are doing in patients with various anemias, normally what happens, you have patients with a congenital anemia. Because they're anemic, they become hypoxic. If you don't have enough red cells, then you don't get enough oxygen to the tissues. If there's not enough oxygen to the tissues, then there's a drive by the kidneys to make stuff called erythropoietin, or EPO, much loved by people who want to increase their oxygen supply to the tissues and ride faster on bikes. So erythropoietin is produced, and that makes a drive to make red cells and hopefully correct the anemia. But it doesn't quite work like that, because as the cells are made, there are things called growth factor inhibitors get expressed on the cells, and that stops the production of the red cells. So if you have a particular disease, you end up with too few red cells. So these drugs come along here at this point and they suppress this GDF11, and the end result you would hope would be increased red cells and correction of the anemia. This slide looks at the soft-receptor and looks at it in more detail, but again, it's the same idea. You're taking the drug and they're binding on the cell surface, and basically the actin receptor type linked to the fragment on the cell surface is trapped, and you end up relieving the blockade of the terminal erythropoiesis. Like in my last slide, the end result is more red cells get made. So these drugs have been shown to be effective preclinically in things like thalassemia and Diamond-Blackfan syndrome, which are congenital anemias, and they're now being taken into trial in patients with myeloproliferative disorders, obviously particularly myelofibrosis with anemia, to see if, using the same mechanism, they would be of use in treating the anemia. And this is a slide showing a patient in an early trial who has been treated regularly with one of these drugs, and you can see that their hemoglobin over time has come up, and that they become less anemic and don't need transfusion all the time. So again, these are drugs that are coming into clinical trials looking for the treatment of myeloproliferative disorders, but in a different mechanism from the inhibiting of the JAK-STAT pathway that we've been talking about previously. Okay, so that's just a flavour of some of the drugs that are in clinical trial at the moment. But let's look at some of the other ways that we may think either now or in the future of providing treatments for myeloproliferative neoplasms. So we always have to come back to the genetics to look at this, and I'm sure you're all very familiar with this idea. We have the human with the cell, we get down to the nucleus and then the chromosome, and then along the chromosome we have the DNA where all the genes are, and that is obviously the backbone of all of our cells. So along the DNA we have the genetic code, and of course, as we've heard already a number of times, in the myeloproliferative neoplasms, we get acquired mutations which produce changes in the genetic code, so that at some point along this DNA you get a change that happens in the cells, a mutated gene. I saw somebody asking what was the JAK2 mutation. This is one of these genes where there's been a change in the genetic code. So the idea is, could you do something basically to edit the genetic code? Could you take out a bit of this code and either remove the bit that had mutated completely, or perhaps replace it with the gene that was normal rather than mutated? And this concept has been worked on, as all these things, for many, many years, and it is now coming into fruition. So I'm going to try to explain it a little bit more on this slide. The idea is, again, what we're actually talking about here is genome editing, trying to change part of the sequence of the genetic code. So again, we have our nice bit of DNA making RNA, but what you do is there's mechanisms, which I'll show you in the next slide, where you can recognize specific DNA sequences, and then you can use this to change, cut out or remove a bit of the DNA that you don't like, and then you end up with a genetic code which has a bit changed or altered or edited. This slide perhaps shows you a little bit more what you can do. Here you have your genetic code, and you can either delete a bit that you don't want, or, in the good computer technology, copy and paste a bit that you do want. So take a good bit and replace it and put it in somewhere else. So it really is editing the DNA. This technology is what everyone is referring to as CRISPR-R, and it is being used both at a research level and at a clinical level all over the place. Everybody, new technology, everybody has a look at it with their disease. The bit behind it depends on the fact that bacteria combat mechanisms to stop invading viruses. And there are therefore these areas in the genetic code called palindromes. Palindromes were something I always learnt at the school. The same forwards as backwards. It's the same backwards and forwards. It was one of those definitions you learnt at school. But with these palindromes are along the DNA, and the technology comes along and uses these clustered, regularly interspersed, short palindromic repeats with the effector, which is caspase 9, and basically recognizes the bits that you want to alter, goes in and either edits the gene either by deleting something or by replacing it. So you can maybe see it a little bit better here in this slide, where here you have the technology to remove and replace a piece of the DNA. So, CRISPR-R gene therapy finally coming to MPNs. Well, it is coming and people are looking at it. But I have a few opinions, which I think are quite interesting, of things that have been put up of where it's going, and then we'll raise questions which I won't be able to answer as to where it's going to go in the future. But certainly, places like Harvard Medical School have established isogenetic human-induced pluripotent stem cells containing CRISPR-R engineered MPN mutations. So certainly in stem cell culture, people can do this to alter the MPN mutations. And by doing that, at least in the cell lines, correct the JAK2 mutation. So it can be done. It's exciting and novel, but there's always a but. It's very preliminary, and I think we have to be aware, while it could work in theory, there are many unanswered questions and safety issues. How do we do this effectively and safely? Because that will be the question that will rise. If you start moving bits of the genome away and taking bits out, what are the effects of that? So the bottom line is the long-term costs, because it's not going to be cheap, this, and the benefits are yet unclear. But it's coming, and I'm sure over the years we will find out more, and this will move on. Other things, how long does this response, how many repeat treatments, all sorts of questions, and cost always comes in, because these things are always hugely expensive at the beginning. But I think gene editing as a technology will come to MPN and right across oncology and other areas of medicine. But it's a long road ahead. So the other thing I wanted to look at for a few minutes, just at this end of this talk, is the immune system, and whether there are any therapies that are coming from immune regulation or maybe will come in the future. So the immune system, I think, is fascinating, and certainly if in malignant practice, actually, if you go back, always you have to think, why has somebody got this acquired disease? It is in some way a failure of the immune system. When I was at medical school, the immunologists were fascinating, but in one way, the immune system fitted into the box that I would have referred to as the too difficult box, because it was very difficult and complicated. And sometimes I think that demonstrates the fact that we don't actually understand. And I think that's still entirely true of the immune system. But progress is happening, and I think a lot more progress will happen. So the immune system is a multi-organ system where we have lymphoid tissue in lymph nodes, the spleen, and the bone marrow. And then at the bone marrow level, we have, as we all know, all these different cells, but there are many cells that are produced by the blood, particularly in the lymphocyte class, but some of these other cells as well that are part of the immune system. We can look at this in more detail, and this is where it always gets more and more complicated, but it starts out with antigens on the cell surface, which are the things that can be recognized and makes one cell different from another. And then there are lots of different types of immune cells that are involved in the response to antigens. B cells and T cells. B cells make antibodies, and the antibodies attack the antigens, but all sorts of other things like T helper cells, T memory cells. You get mumps when you're a baby. You have memory cells that remember that, and if you ever get exposed to mumps again, make more antibodies so that you never get that disease again. That's why vaccination works, because you don't get the disease if you've got exposed to the antigen and make antibodies. But there are all sorts of other activated T cells which are part of this response. And when we take this further in immuno-oncology, in the response to tumours, the immune system has a lot of roles to play. Here we have a tumour which makes lots of antigen, and then we have various cells in the immune system, like antigen-presenting cells, who look at the antigen and make it ready to be attacked by the T cells, and various ways at the cell surface. Here we have an activating antigen-presenting cell, and various mechanisms at the surface to get the T cells so that they can look at the antigen from the tumour cell and attack it and hopefully destroy it. That's what should happen. But things can go wrong with this, and by definition almost, by the time somebody has a tumour of any sort, something has gone wrong because the tumour has been allowed to grow. There are various bits of this that are involved in more detail at attacking tumours. There are cytotoxic CD8 T cells, which are involved in this mechanism of the lymph node or the immune system attacking the tumour. And then we have other cells like CD8, further T cells, which are also part of this process. So there's a whole complicated process of various types of T cells which are part of the process to attack and kill cancer cells. And this slide's a bit wordy, but I need the words to take you through some of the bits of this. There's a particularly process on the cell where there are immune checkpoints, where there are parts of the process where the cells can check and see, is this a tumour and can we get rid of it? And these are the PD1, or programmed cell death. But the problem is the tumour cells often take advantage of these checkpoints to escape the immune system. And then there are further proteins come along and they attack, bind on PD-L1 to the PD-1 and try to help the T cells fighting the process so that they actually work and get rid of the tumour cells. And of course, what is happening now is drugs are being developed at these immune checkpoints to try and increase the process so that the T cells will then get rid of the tumour cells. And one of these is nivolumab, which can block the tumour from deactivating the immune system. So basically comes in here where the PD-1 is and blocks this happening so that then the T cell then kills off the tumour cell. And these drugs are actually licensed and are being used in various different types of oncological diseases, melanoma and others like that. This is probably a further slide, very complicated, but again showing you the same point where the novulimab fits on as an antibody blocking the PD-1 so that then the T cell will destroy the tumour cell. And there is now a phase 2 trial of this drug in myelofibrosis. So again, this is another way of taking a totally different mechanism, checkpoint inhibitors, into manipulating the immune system into the treatment of myeloproliferative diseases. So this drug is in phase 2 trials in MD Anderson. So the final thing I wanted to mention, because it's the hot news, are CAR T cells. And again, this is immune regulation. These are looking at chimeric antigen receptors, again looking at the receptors for the antigens. So every cell has antigens, looking at the receptors and manipulating them so that they can be used to attack the particular type of cell. There are various types of this. It started off in the first generation just with an antigen recognition domain and various signaling bits. But we're now at the point where this can be used, and basically what happens is T cells are collected from the patient. Then, when you're making CAR T cells, you end up taking a chimeric antigen receptor, a sort of manufactured receptor to whatever antibody you want, put that on the, that is something that will recognize the antigens on the tumour cell surface. You take these T cells and in vitro, the engineered T cells, you multiply them so that you get lots and lots and lots of them, and then they can be given back to the patient. And that is CAR T cells. They have been now produced against particular B cell antigens and have proved very effective in the treatment of certain very specific lymphoid leukemias, and have now been licensed. The drug is called Tisagen-Lucel. I have to keep it on a bit of paper because it's the only way I'm going to get through that. But that is now present and useful in treatment. Where it will go, I don't know. I know people are starting to look at it in myeloid cells, and there's been some work in solid tumours, not very successfully. But again, it's a totally different way at looking at treating diseases, and I'm sure over the next few years it will go on. So I'm done there. I've covered some new drugs and some new immune therapies. Thank you very much.